Okay, welcome. Today we shall continue to see translation. How protein is synthesized in both eukaryotic and the prokaryotic cell type. Before I proceed to the detail of our discussion, if you are not a member of Nigi Academy, subscribe the video. If you are already a member of Nigi Academy, share and comment on this video. Let us continue. Translation is the process by which information contained in RNA is used for synthesis of protein. You know, in transcription, the base sequence in DNA changed into that of RNA. There is a production of RNA. That RNA might be messenger RNA, transfer RNA, ribosomal RNA, or another kind of RNA. From that all kind of RNA, messenger RNA is very, very important. It is a coding RNA. It used to code for protein. It codes for protein. So when we say translation, it is the messenger RNA sequences translated to protein sequences. The sequence of amino acid in protein synthesized is determined by nucleotide base sequence of messenger RNA. It used to protein production. The information coded in 4 bases found in messenger RNAs translated into the instruction encoded by 20 amino acids used in formation of protein. Mind you, there are 20 amino acids. There are 20 transfer RNA. So in translation, the information encoded, the information contained in messenger RNAs used to determine the kind of protein produced, the kind of amino acid produced. Okay. There are different terminologies like a genetic code. Genetic code is, it's nothing, it is triplet nucleotides. There are 3 nucleotides or a triplet base sequence in messenger RNA that act as a code for amino acids. It is in the form of codons or genetic code. So this genetic code is, it's regarded as dictionary of nucleotides. There are 4 nitrogenous bases: adenine, guanine, thymine, cytosine, or a uracil. By these 4 may produce 64 different combinations. That means 4:3. It is a triplet. 4 is a nitrogenous base. So it gives us 64. From that 64 different combinations, 61 are used to code for amino acids. The rest 3 are stop codons: UAA, UAG, and UGA. These are stop codons. While protein is produced, if these stop codons come, it stops the work. That is why its name is termination codon. Just like stop codon, there are starting codons which code for methionine, AUG, or GUG. It is start codon. It is start codon. What are the characteristics of codons? The first characteristic is universality. When you say universality, the same codons are used to code for the same amino acid in all living organisms. There is one exception. For example, AUA is coded for methionine in mitochondria. Again, it may code for isoleucine in cytoplasm. Except this, the rest are universal. The same codons are used to code for the same amino acid in all cells, in all cell. The other characteristics is specificity. It means that it is specific. Particular codons always code for the same amino acid. For example, AUG code for methionine. UAA code for stop codon. GUG code for methionine, not isoleucine, not valine or another kind of amino acid. So it is very, very specific. Again, it is non-overlapping. It never overlaps. It reads from fixed point as a continuous base sequence, but never overlap. Again, it is degenerate. It means that most of amino acids do have more than one codons. More than one codons. For example, UUU, UUC, it is a codons for phenylaline. UUA, UUG, it is code for leucine amino acid. These 4 codons used for serine. So it is a degenerate. It is a degenerate. Most amino acids do have more than one codons. More than one codons. That is why it is a degenerate. There are 64 combinations. From that 64, 61 are stop codon. The rest 3 are stop codon. 61 are code for protein. 3 are stop codons. Okay. The question is, how translation occur? How translation is happening in both prokaryotic and eukaryotic? Actually, the process of protein synthesis or translation occur on the surface of ribosome. On the surface of ribosome. In case of prokaryotic cell, there is only one type of ribosome, which is located in cytoplasm. But in case of eukaryotic, 2 ribosomes. One may be freely in the cytoplasm. The others may be on another organelles like reticulum endoplasmic, mitochondria, chloroplast, and so on. So anyway, the process of protein synthesis is carried out on this machine, on this machine. Okay. Actually, during bacterial translation, messenger RNA should be laid on the surface of ribosome anyway. Surface ribosome. Okay. Then they form a complex. Finally, largest amino will come and they form initiation complex anyway. Let us see in detail of this anyway. So ribosome contains 3 sites: A site, P site, and the E site. P site is the site at which they form polypeptide. They form polypeptide chain. A site is the site at which new amino acid transfer RNA bind to the messenger RNA. Mind you here, messenger RNA carries coded information. Coded information. You know there is transfer RNA. Transfer RNA do have anticodon. That anticodons are very important to recognize a codons which is carried by messenger RNA. So here at A site, amino acid transfer RNA binds to the messenger RNA. Mind you here, the matter of protein synthesis is, it is only not a role of transfer RNA or ribosome, it's the role of many components. There is transfer RNA, messenger RNA, again a ribosome. So, amino acid transfer RNA binds to the messenger RNA. Here, there is a small ribosomal RNA. Here. Then after that, at the P site, transfer RNA carrying nascent polypeptide chain. Here, the joining of amino acid to the chain occur here. They form a peptide. They form a polypeptide. E site is, it is exit site. is the site at which uncharged transfer RNA leave this area. It removed. It passed away. So it is the site by which uncharged transfer RNA leave the ribosome after it gives free amino acid to growing this polypeptide. That is a site on a given ribosome. A site, P site, and E site. Okay. What are the stages? There are 3 stages anyway. The first stage, it is initiation, elongation, and termination here. What happens here, in initiation, there are 3 or 4 components. Ribosome, maybe larger or smaller subunit, messenger RNA, which carries coded information, initiator transfer RNA, which carrying the first amino acid in protein, particularly meat or methionine. Then after these all come and join together, they form initiation complex. That is what happened during initiation. Actually, first, transfer RNA carrying methionine attached to the small ribosomal subunit. Together, they bind to at the 5' end of messenger RNA. Okay? There is a 5' end and a 3' end. First binding occurs at the 5' cap. You can watch my video of post-transcriptional modification. So that complex binds at the 5' cap. Then it moves toward this 3' end direction. Then stopping when the reach start coding, AUG, which code for methionine. Look at here. First, initiator transfer RNA, small ribosomal subunit, binds at 5' cap. Then it walks toward these 3' ends. While it is walking, after it reaches start coding, they stop. Then larger subunit of ribosome come and bind to them. This is a larger ribosomal subunit. This is smaller. Smaller ribosomal subunit already binded here. Finally, large subunit will come and bind. Then they form initiation complex. So it means that it is initiated, just it is started. Actually, this is in case of eukaryotic. In case of bacteria, the situation is a little bit different. Here, the small subunit of ribosome does not start at 5' end because there is no cap, there is no pollination in case of bacteria. There is no post-transcriptional modifications. Instead, it attaches directly to a certain sequence in messenger RNA. That sequence is we call child organo sequence. GG, A, GG. So it binds at this area. We call it child organo sequence. There is 16 ribosomal RNA. That 16 ribosomal RNA bind at this region. That region is we call child organo sequence. That is right in case of bacteria. Then they move here. They form initiation complex. Then they start. Here is initiation complex, larger subunit, smaller subunit. So in such a way, they start. That is the situation in case of bacteria. The question is, why use child organo sequence? Because the bacterial genes are often transcribed in a group. That is operon system. There are many structural genes that are regulated under the one promoter. That is polycystronic. Many genes controlled under one promoter, which is operon system. So one bacterial messenger RNA can contain coding sequence for several genes, structural genes controlled under one promoter. That is operon system. No such issue in case of eukaryotic. Therefore, Short organelle sequence marks the start of each coding sequence, letting the ribosome find the right start codon for each gene. That is how it is started. Elongation is the same in all. Actually, in elongation, once it is initiated, the first methionine carrying transfer RNA start out in middle slot of ribosome. We call it P site, already I mentioned. Then the fresh codon is exposed in another slot at A site, that is a new. The A site will be the landing site for next transfer RNA. Once the matching of transfer RNA has landed in A site, it is a time for action. That is the formation of peptide bond that connects one amino acid to another. They form polypeptide. Actually, amino acid do have two terminus, amino terminus and carboxyl terminus, N terminus and carboxyl terminus. So one amino acid attach one another in N or T terminal. In such a way, it forms a polypeptide. Then, after that polypeptide is synthesized, it has to stop. How to stop? That is what we call a termination. That happens when the phase stop codon in the messenger RNA, UAA, UAG, UGA. If these stop codons enters at A site, they stop synthesis of amino acid polypeptides. These stop codons are recognized by protein release factor, which is fit neatly into P site, though they are not transfer RNA. This release factor is stimulate release of peptide from ribosome. It stimulates release of that complexes. You know, there is two ribosomal subunits, larger and smaller, transfer RNA, messenger RNA. So this release factor stimulates peptide from ribosome. Once that peptide is released, the rest again will be released. All the complex messenger RNA subunit to ribosome, transfer RNA now it's released, so they are ready for the next job. The question is, our polypeptide now has all its amino acids. Does that mean it is ready to do its job? Is that protein active? Is that protein functional? The exact answer is, it's not necessarily, because that protein is not active. Not active. It needs some editing during and after translation. There should be modifications like folding, proteolytic cleavage, itin splicing. Chemical modifications are very important. These all modifications are what we call post-translational modifications. These are necessarily important to make the produced protein active. Then the protein localization maybe takes place. That is how translation is terminated. For example, folding of protein into some tertiary structure, proteolytic degradation, some portion of precursor molecules should be removed, itins, which is like that of introns, should be removed, covalent changes, phosphorylation, hydroxylation, glycolation, these are very, very important. Thank you very much.